Ladies and gentlemen, welcome to our MCQ biology revision, and in this particular class we are going to look at the 2023 exam paper, of which this is Bio 1400. So there are about 100 of questions. There are about 100 of questions, but we can't finish all the 100. So today I'll do 25 of them. Then in the next class I'll do 26 up to 50, and then I'll go and start doing Bio 1401 and 1412. All right. So with that being said, let me quickly start up our first question, which is, which of the following is not a property of water? So for you to know much details about water, what water is, go and check up our lecture class on our YouTube platform, and you're going to know much about what water is and many things you need to know about it. So the first one, it has high surface tension. This is correct. It has high heat capacity. It's correct. It requires a lot of heat for it to be converted from a liquid. Yes, which is correct. It is a non-polar solvent. No. And then this one is correct. So the answer for this one was simply 4, which is it is a non-polar. So we need to understand that water is a polar solvent, and it being a polar solvent, that simply means that it's able to make the hydrogen bond within itself, and of course it can dissolve other polar molecules. Then nucleotides are composed of dash. So for this one, I will make a lecture class on what the nucleotides are, and also what nucleic acids are. So please make sure that you attend the class. I'll explain about that. So the first one is ribose and proteins. Ribose and proteins. And then this is wrong because of protein there. Deoxyribose and phosphate, deoxyribose and phosphate, which is wrong, like that. And then we have sugar, nitrogenous base and protein. This is wrong because it's not protein. Then nucleoside and phosphate, this one is correct. So a nucleotide, it is made up of the nucleoside and the phosphate, the nucleoside and the phosphate. So this is the correct answer for the for this particular question. Okay, let's move on. When I reach on number 10, I'll allow you guys to ask question. Which of the following structure is found in both prokaryotic and eukaryotic cells? Please go and check out the cells on our YouTube platform. I've explained this particular part. So one is saying they both contain Golgi apparatus, which is wrong. Endoplasmic reticulum, which is wrong. Mitochondria, which is wrong. And the other one is nucleus, and then the ribose. So the answer here is ribose. The reason why we can't include this one, that one, and that one, it's because these are organelles. Okay, these are organelles. So mostly the prokaryotics do not have the organelles. Then the nucleus, most of the people would be tempted to pick the nucleus. Yes, you might be correct, but this thing is more like it is not definite. I can use such a word. It is more, the DNA is not compressed, because the DNA of the prokaryotic is found within the cytoplasm. So for this one, a lot of people picked 4, but the answer is just the ribosomes. Proteins are modified in the dash of the eukaryotic cell. So here we have smooth endoplasmic reticulum, Golgi apparatus, ribosome, nucleus, nucleolus. So this question is coming from DNA translation. I will send notes on this. So the answer for this one is simply where the modification takes place is simply in the nucleus. That's where DNA the protein modification takes place. So proteins are being modified in the nucleus. So one thing that you have to take note of is that most of the RNA, the DNA, they all originate from the nucleus in eukaryotic cells. So the nucleus, it acts more of a center of everything, and determine what happens to any metabolic reactions in our body. So I hope that is clear to everyone. Okay, so let's move on and look at the next question. The next question is, why is the amino group in protein polar? Why is the amino group in proteins polar? Okay, this is a very interesting question. But before I look at this question, you guys need to understand that proteins are polar molecules because of a certain bond which occurs in them. So the answer on that particular question, it is simply 2, because of nitrogen is more electronegativity than oxygen. I think this one you find it when I was explaining bonding, specifically when I was talking about the polar covalent bond. The video is there on our YouTube channel. Go and check it out. You're going to find the more explanation. So due to the difference in electronegativity between nitrogen and hydrogen, these guys, they have a dipole moment, meaning one side is partially positive and the other side is partially negative. So this side is partially positive and this one is partially negative and the other one is partially positive, just like the way it happens in water. Number 6, identify the incorrect statement about enzymes. So when you talk of enzymes are just proteins that speed up a chemical reaction, or they reduce a chemical reaction. And most of these enzymes, they are normally protein in nature. So 1, they are much larger than their substrate, which is wrong. Only a small portion of their structure is directly involved in catalysis. Okay, we can give it a question mark. The lock and key model explains the enzyme specific, which is this guy is also wrong. I'm trying to pick what is incorrect here. So when I say wrong, I simply mean that it is correct. Enzyme work by raising activation energy needed for the reaction. enzyme work by raising the activation energy for the reaction. Okay, we have to understand one thing on this. Actually, this is the answer on this particular question. So for an enzyme to speed up a chemical reaction, it doesn't increase the activation energy. Instead, it normally reduces the activation energy, because this activation energy is the energy which is making a chemical reaction to take place. So for that chemical reaction to take place, to take place at the fast speed, automatically it has to be what? It has to be reduced. So this was the incorrect answer on this particular question. Okay, let's move on and look at number 7. Identify the structure that is not found in prokaryotes. Identify a structure that is not found in prokaryote. I think here this question is related to that question, so the explanation I gave there is the same explanation you can get from there. So I'm sure plasmid, most of you might get confused. Cell membrane, cell wall, ribosome. So cell wall, some prokaryotic cells, they have cell walls, because when you talk of prokaryotic cells, you can talk about a bacterium. Okay, you can talk about a bacterium. There are some bacteria that have a cell wall. There are some bacteria that have a cell wall, and these bacteria are able to transmit the diseases through the same cell wall by attaching itself to the host cells. I'm not going to go much in detail on that because that is more of microbiology. So the answer here is mitochondria, because this is an organelle which is double membrane. Which of the following is not true about the transfer RNA structure? So this is tRNA, and when you talk about tRNA, where you can get it from, you can talk about the types of RNA, and this topic is coming from transcription. So this topic is normally coming from, this question is coming from transcription. So when you talk of that, the types of RNA, if you go much in detail, you need to understand that for the D arm, the structure of RNA, so the structure of RNA is that it has a D arm, it also has a T arm, then it also has the anticodon arm, and it has the amino receptor arm, and the CCA sequence is found at its 5-cap end, which is wrong. It is found on the 3-cap end. Yeah, it's found on the 3-cap end. So the answer here is simply that. So basically, when you talk of biology, you need to do much of the research. If you look at number 8, none of your lecturers notes have this part. None of your lecturers notes has this explanation. Even the A-level books, they don't have this. The A-level books, they don't have this. This question is coming from biochemistry. It's coming from biochemistry. So only someone who has done biochemistry is able to understand this particular question. So do much of the research. The next one is select a statement that is true about the plasma membrane. So this plasma membrane, you need to understand that it has a lipid bilayer. That's one of the things that is there. It is selectively permeable, selectively permeable. So this is the basic information most of you have, but one thing that you need to understand that this same plasma membrane has also another type of lipid, which is known as the cholesterol. So the cholesterol, it's a lipid which normally determine the fluidity and rigidness of the plasma membrane. So for this one, which statement is true? So we have all living cells are surrounded by a plasma membrane. All living cells are surrounded by a plasma membrane. Then cell receive chemical messages from the environment through the plasma membrane. Plasma membrane have protein channels through which nutrient for the cell can pass. Then 4, cholesterol is part of the plasma membrane, and 5, all of the above. So the answer here is simply all of the above. So there are some situations, this is just a tip which I'm giving you. There are some situations whereby you only know 2 options, the other options you don't know them. Then you find an option which is saying all of the above. Automatically, if it's saying all of the above, according to you guys, most of you guys, I know this one you are able to remember. You are also able to remember this one because I've told you that there is another lipid which is known as cholesterol, and this cholesterol, it determines the fluidity and rigidness of a cell membrane. So if you can remember that one, and these two are giving you a headache, you can simply go with all of the above. Then this option 2, you can easily know much about it when you look at physiology of a cell. So if you look at physiology of the cell, you'll be able to understand that the cell is made up of the extracellular fluids and the intracellular fluids, and there is a communication between the intracellular content and the extracellular content. Yeah, that's why there is something like the sodium potassium pump, something like that. Then the other one is plasma membrane have protein channel through which nutrient for the cell can pass. This one is correct. A prokaryotic cell lacks dash. Okay, here now we are being asked to look at what a prokaryotic cell lacks. So the answer here is nuclear envelope. I think I've explained something like the DNA content of a prokaryotic cell is not really found inside the nucleus, but it's scattered all over the cytoplasm. The last one for this section is, which of the following is true about enzyme cofactor? So I'll explain much about enzymes and the cofactor later on. So the first one is cofactors are always proteins. Then enzyme cofactors are always protein. This is no, this is wrong. Enzymes can function without the presence of cofactor. Okay, cofactor are primarily involved in stabilizing the enzyme's tertiary structure. Cofactor are essential for enzyme catalyst. Cofactor are carbohydrates in nature. This is wrong. This one, no. So we are having enzyme can function without the presence of cofactor. So if you look at the options which are remaining, then cofactors are primarily involved in stabilizing the enzyme tertiary structure. This one is wrong again. This one is wrong. because basically there they are trying to describe a proteins. Then cofactor are essential for enzyme catalytic activity. Cofactor so this one again, it is wrong. This one, it is wrong. This guy here, it is wrong. Okay, this one is wrong. Why is it wrong? When you look at cofactor, they normally act as competitors for enzymes. Cofactors, they normally act as competitors of enzymes, so you can't they can't really be that important. So enzymes can function without the presence of cofactor, which is correct. Yeah. Okay. The next question is, bacterial cell wall is made up of dash. So this again is taking back to the cell type. So the answer for this guy is just the peptidoglycan. This is the answer. This one, more of explanation on this, you can study microbiology. So when you study microbiology, you know much about the content of the cell wall of the bacteria. And this peptidoglycan, they are normally made up of the protein. They are normally made up of proteins plus carbohydrates. They are made up of proteins plus carbohydrates. And in such cases, you find that in some bacteria, these same guys, they are used to cause some infections. The number of dash is equivalent to the atomic number. I think this is easy. Everyone can get that one correct. We can move on. Which of the following is a normal pair of nitrogenous bases of DNA? Which of the following is the base pair of nitrogenous bases of DNA? So guanine and cytosine, I won't even waste time. This is the correct answer. In the electron configuration for the oxygen atom, how many electrons occupy the second energy level? So oxygen, which is O, has 16 and 8. So you won't be given the periodic table, so some of these things you need to know them. So you know this part, it will be occupied by 2 electrons. Then the other part will have, let's say, 1, 2, then you have 3, 4, and then you have 5. Yeah, there are 6. So 1, 2, 3, 4, 5, 6. They are in the second shell. This is the first shell. Okay, yeah, the first shell. So the first one will have 2, the second shell will have 6 of them. So the answer here is simply 6. Which of the following is not a function of the bacterial cell? Which of the following is not a function of a bacterial cell? I feel this section is full of cells and DNA and also enzymes, and I've seen atomic structure plus water. So in short, you need to balance up all the topics. So number 16, the answer here is simply give shape and rigidity to the cell. Yes, of course, it is okay. Protect the cell from rupturing. It is okay. Which of the following is not a function of the bacterial cell membrane? I feel they left something like cell membrane. I feel they left such, because yeah. Then surrounds the plasma membrane. Okay, we have is the site of action for some antibiotics, and then we have contains genetic material. Which of the following is not a function of the bacterial cell? Bacterial cell. Give shape and rigidity to the cell. Okay, fine. Should it be cell wall? Okay, I feel if this, then this has to be cell wall. It was incomplete. The question was incomplete. The reason why I have put cell wall is that I'm looking at option 3. I'm also looking at option 1. If option 3 is correct, then option 1 will also be correct. Okay, then protect the cell from rupturing. This is also correct. So that simply means that this question, there was a correction of putting a cell wall. Then is the site of action for some antibiotic. Remember guys, when I was talking about the peptoglycans, and also when I talked about the cell, I said in some cells they are able to transmit some bacterium due to the presence of the cell wall, and that cell wall normally has a capsule. Okay, so these antibiotics can be used on the cell wall just by understanding the cell, but I can't go much in detail. That is also microbiology. So we already know that only the nucleus has genetic material. In the atomic nucleus, the attraction of electron to proton is by the dash. So I think when you read the notes I gave, you're going to find out that the attraction between the electrons to proton is by the electromagnetic force. The sucrose is non-reducing sugar because of the presence of the dash glycosidic bond. If you watch my tutorial video, which is carbohydrates part 2, carbohydrates part 2, when I was talking about disaccharide, I clearly emphasize on you guys knowing the type of bond which is in sucrose. So for this one, which is number 23, the answer is simply 5. This is the answer there. Sucrose 1 to 5. 1 to 6. Am I sure on this? Okay, yeah. Beta 1 to 6 glycosidic bond. The next one is number 24. The next one is number 24. So number 24 is still on carbohydrate. Number 24 is still on carbohydrate. Which of the following is not a function? Which of the following is not a function of carbohydrates in living organisms? Let me just go back there. I feel this is so close. So it being a sucrose, what we can state is actually this question had a problem. So there is this one and that one. Both of them are supposed to, one of them is supposed to have the answer, which is supposed to be alpha comma beta 1 to 2 glycosidic bond. So there was there was an error when it came to that particular question, because you need to understand that glucose, which is taking part in what? In formation of sucrose, is alpha. So alpha glucose. So you have alpha glucose plus C, beta fructose. Alpha glucose plus beta fructose, they are going to form what? Alpha beta, alpha beta, and then you have 1 to 2 glycosidic bond. So yeah, this is the correct bond which was supposed to be picked. But in such cases, you can ask for a correction, or you can pick 2. Personally, I can go with 2, though it is not the right one. Which of the following is not a function of carbohydrate in living organism? So energy storage, which is correct. The other one is instant energy, such as glycogen. If you look at glycogen, production of roughage in the human digestive system. This one, you talk about the fibers. This one is about the fibers. Okay, and then the other one is structural support. So if you look at structural support, then we're simply talking about cellulose. Genetic storage, which is wrong, because this is nucleic acid. Water is able to expand when it freezes because of dash. So here the answer is simply the formation of rigid hydrogen bond. This one you won't find it in the notes. This one, it's not in your notes, but what you can do is, the more you go through these papers, the more you know about them. So ladies and gentlemen, thank you very much. For now, I'm going to allow you guys to ask questions. So for those who are in class right now, you are free to ask questions from the first question, and for those who may want to ask later, you can WhatsApp me on those lines.